Good morning, everyone. It's my pleasure to welcome you all to today's PhD seminar, Viva seminar of research scholar Mr. Sharfuddin Mohd. We are deeply honored to have with us an eminent expert, Dr. Mohinder Singh Bedi, professor at Guru Nanak Dev University, Amritsar. Currently, Dr. Bedi serves as Director of Global Ranking and International Collaboration Cell and is professor in the Department of Pharmaceutical Sciences at GNU. With over 27 years of rich experience in teaching, research and administration, Dr. Bedi expertise spans pharmaceutical chemistry, medicinal chemistry and analytical chemistry. His outstanding contribution to the field are evident through the leadership roles at GNU as well as various prestigious national and international positions. Dr. Bedi has successfully completed more than 15 major research projects and has guided over 12 PhD scholars and 40 master students to their degrees. His profile research output includes more than 140 publications which have garnered over 4,600 citations, reflecting an impressive H-index of 37. It is an honor to have Dr. Bedi with us today as the expert seminar for this important milestone in Sharfuddin academic journey. Sir, we warmly welcome you to our campus and thank you for gracing this occasion with your presence. With your permission, we would like to commence the presentation.
Good afternoon to all. Myself, Sharfuddin Mohd. I am presenting my PhD final Viva presentation on my topic synthesis and antidiabetic investigations on thiazole derivatives under the supervision of Dr. Vikas Sharma and co-supervision under Rakesh Sharma, sir, School of Pharmaceutical Science, Lovely Professional University. So these are my contents for my presentation. First is introduction. Basic introduction on the diabetes, literature review. Based on the literature review, we have frame a rationale, aim and objective, and work plan. Our work plan is divided into two parts. One is series 1, which is concerned to NT1 and NT18, which is about 18 compounds were synthesized, and series 2, QT1 and QT8. First, introduction. Diabetes is one of the chronic metabolic disorder, which is concerned to elevated blood glucose levels in the body. This diabetes is not only concerned to the blood glucose evolution, difference in the glucose levels, but it is also associated with the different parts in the body. Like this is a present scenario as per the International Diabetic Federation. So present 11th edition was released. So this is a scenario. Worldwide, the present 588 million people are suffering. Maybe this is going to be predicted 45% increase in up to 2025. And very important point to be highlighted in this is that diabetes caused 3.4 million deaths in last 2024, means every 6 seconds one death, so which purely alarms. Diabetes is categorized into 4 different types: type 1 diabetes, type 2 diabetes, where type 1 is concerned to autoimmune disorder where the insulin production is totally absent; type 2 diabetes, gestational diabetes, and monogenic. Our focus is on type 2 diabetes as it concerns 90-95% of diabetes cases. And these are the chronic complications which are associated to diabetes, which is not treated for the longer duration. And the management and the treatment of diabetes, one of mainly lifestyle changes is the first parameter, and next we going with the monitoring regular glucose levels. This is the second parameter, and we have different types of medications. Type 1 diabetes is concerned to insulin therapy, which is only the one option for the insulin treatment, type 1 treatment. And for the type 2 diabetes, we have oral hypoglycemic agents. Like oral hypoglycemic agents, sulfonylureas generation 1, generation 2, first generation and second generation, metformin, which comes under bigonide derivatives, glitazones, which is having a very concerned scaffold, which is called thiazolidone derivative, and pancreatic alpha amylase and intestinal alpha glucosidase inhibitor, acarbose, maglitol, voglibose, magnetol are some examples. These are the oral hypoglycemic agents which are approved by the FDA for the present treatment for the type 2 diabetes. But there are some serious side effects which are concerned to the present therapy, present drugs, like most commonly diarrhea, and the most common point is hypoglycemic conditions. Due to this oral hypoglycemic drugs, the hypoglycemia is one of the parameters. So, scaffold point of view, we have selected thiazole derivative, which is commonly reported for vitamin B1 and for having a various biological activities. In this, the thiazolidone derivatives are used thiazole derivatives which are having a thiazolidone ring, which is having 2,4 diones with nitrogen and sulfur atom at first position sulfur and N3, and these scaffold is mostly preferred for the glitazones which are approved by the FDA, like example siaglitazone, ripaglitazone, nataglitazone, which are in the clinical applications. Pancreatic alpha amylase and intestinal alpha glucosidase. This is the enzymes which are mainly targeted for the management of diabetes mellitus. Like alpha amylase pancreatic alpha amylase have ability to inhibit the conversion of starch into glucose. As a result, the glucose metabolism is going to be stopped. And where in concern to alpha glucosidase, the inhibition of sucrose conversion into the glucose is going to be inhibited. Literature review. So we worked mainly on scaffold strategy, different thiazolidone derivatives with heterocyclic compounds, like furon and thiophone is conjugated with thiazolidone, and thiophene is conjugated with thiazolidone, which is reported for the anti-diabetic activity, and coumarin based derivatives are conjugated with thiazolidone, chromone based derivatives which are conjugated with the thiazolidone, isoxazole based derivatives, isoxazole and oxadiazole based derivatives, which is reported for mainly alpha amylase and alpha glucosidase activity in combination with thiazolidone and the heterocyclic moieties. Benzoxadiazole derivatives, which is also reported for the alpha amylase activity, and benzoxazole and benzothiazole derivatives, like quinoline based derivatives, which is reported for in vivo report studies, in quinoline based studies, naphthalene based thiazolidone derivatives, and phenothiazine and aryl based derivatives, and even pyrrozole based derivatives. And these are the thiazolidone drugs which are in clinical use, like we generally call them as glitazones, like C-glitazone, rosiglitazone, piaglitazone, nataglitazones. These are the drugs which are in the clinical use. This is the publication on introduction and the literature review. Recently we got a publication on obesity medicine, which is indexed in Scopus, having SGR 0.430. In this we have covered maximum understanding on diabetes, purely extensive work on diabetes, its complication, novel targets, and advances in treatment strategies. Rationale, aim, objective, and work plan. So presently, by seeing our extensive research literature survey, So we have concluded, we currently explore the option of finding the more potent and the less toxic compounds with the treatment of diabetes with the better ADMET profiles using thiazolidone analogs. So this is a rationale. Rationale is generally we have divided into three main components. One which is concerned to the drugs which are already reported on the thiazolidone based derivatives, and these are the drugs which are under clinical applications, and this is our main rationale. So in this, the green color, which is generally considered as a thiazolidone, this is a core moiety, central core moiety. To this central core moiety, based on the literature, literature is very clearly suggests that on the thiazolidone moiety, C position and the N position only used for the substitution concern. So C substitution, generally we prefer the aldehyde with thiazolidone, we generally call clovengenol condensation reaction, and N substitution. On nitrogen, there is a chance to do the substitution. So based on this literature, C5 substitution is preferred and the N3 substitution is preferred. And C, on the third position there is a carbon, and on the third position, fifth position there is a carbon, and the third position is a nitrogen. So based on this, we have used quinoline and the naphthalene as an option. And on N substitution, we have used various aryl, alkyl, halyl, haloalkyl and alkyl groups for the different derivatives. So based on this, we divided into series 1 and series 2. One is naphthalene based thiazolidone. Generally we prefer the word NT, which preferred naphthalene thiazolidone derivatives, naphthalene based thiazolidone derivatives. And second series is quinoline, quinoline thiazolidone based derivatives. So we have framed, the main aim of my work is synthesis and anti-diabetic investigation on thiazole derivatives. First objective, designing of novel thiazolidone derivatives, thiazole derivatives as a potential anti-diabetic agents. Second, we synthesized and characterized the design moieties, and in vitro screening of this synthesized moieties. This is the work plan of work for our work. First, we have designed the moieties. Just now I have proposed the rationale, how we have gone for the rational designing. Second thing, synthesis and characterization. Here we have done purification, melting point determination, FTIR, mass spectroscopy, HNMR and C13. In HNMR we have gone for HRMS analysis also. And in vitro studies, pancreatic alpha amylase, alpha glucosidase inhibitor, and antioxidant activity, because ROS is one of the parameter which is induce the diabetes concern, oxidative stress in the cells. So we have also studied the antioxidant activity of the thiazolidone derivatives which we proposed. And we have validated this in vitro data with in silico studies like docking, GASBA analysis, molecular mechanic GASBA analysis, and also simulations, dynamic simulations. Materials and methods. So this is the methodology which is used for the investigating the anti-diabetic activity. And DPPH antioxidant activity, DPPH proposed literature is used for the detection of anti-diabetic activity. Second, computational investigations. On computational investigation is generally used for the validation purpose. Then this molecular docking with Autodoc Vina and retrieval of proteins from the database, and the results were analyzed by the BioVia Discovery Studio. And MMGBSA prime module we have used, and molecular dynamics desmoid 200 nanoseconds. We preferred 100 nanoseconds is a one parameter, and we have also gone with advanced 200 nanometers. And molecular properties with ADME properties with SysADME and MolSoft web server, and Osiris property explorer is used for the toxicity prediction. Results and discussion. Series 1, generally I have discussed early, we have divided our work in two series, series 1 and series 2. Series 1, here we have synthesized and characterized the design compounds and in vitro antidiabetic and in antioxidant investigation and in silico validation. This is the proposed scheme for the series 1, where first we have used different aldehyde, naphthalene concerned aldehyde and reacted with the thiazolidone. Generally here we have used clovengenol condensation reaction, and the double olefinic linkage is formed in between the thiazole and naphthalene. And for this naphthalene we have preferred, next step is N substitution, N substitution in the presence of alkyl bromides, various types of alkyl like alkyl, haloalkyl and aryl bromides. And N substituted naphthalene based thiazolidone derivatives are prepared. These are the data of physical information, data about molecular formula. We have prepared 1, 2, 3 up to 18 compounds. Molecular formula for the synthesized compounds, molecular weight, melting point for the individual samples. We have verified with literature, yield of different compounds. And this is a characterization data for one of the synthesized compounds, preferred first compound. Molecular mass is 413, color yellow solid powder, yield 84%, melting point is around 225 to 228, and molecular mass is 414. And this is HNMR spectra for NT1, which is characterized here. Main preferred is when we see olefinic bond between this, here the hydrogen is generally, this hydrogen is preferred in aromatic region due to the double bond, and these two electrons are seen at this position 4.97. In vitro antidiabetic and antioxidant study for first series, series 1 for 18 compounds. This is the report of antioxidant activity where NT17 and NT7 are having very good anti-alpha amylase activity. And intestinal alpha glucosidase inhibition activity for the series 1 compounds. In this same NT7 and NT17 having a good anti-alpha glucosidase activity. And we have compared with acarbose as a standard drug. This is the NT7, sorry for this, topological error please, and NT17, so acarbose is standard. And in vitro antioxidant activity for the NT series. Here NT7 is having a good antioxidant activity and NT17 is also having a good antioxidant activity. This is NT7 and 17. And it is compared with acarbose. In silico validation, these are the 2D and 3D interactions. We generally use Autodoc Vena for this detection, validation. Here we have one hydrogen with GLY306 and it is also verified with the literature. And this is a 3D picture of interaction, and this is the aromatic surface area and hydrogen bond interaction and SAS, solvent accessibility surface area. And this is for 17, N17. ARG315 having a hydrogen bond, and this is a 3D interaction and aromatic surface, hydrogen bonds and SAS surface. And also we have performed the ADME studies for the compounds, for 18 compounds, and we are taking acarbose as a standard. And here we have performed the drug likeness properties with various parameters like Gauss, Weber, Egan, Lipinski parameter. So they are in the limits as per the ADME parameters. And also studied the toxicity studies, mutagenic, tumorogenic and reproductive effect or irritant effect, and all the preferred compounds which are synthesized, except one which is in red color, red. This is somewhat irritant, and remaining all the compounds having a good profile against toxicity. This is a publication on series 1 in which is index in Scopus and SCA, having an impact factor of 2.5, cell biochemistry and biophysics general. And results and discussion on series 2 to measure the difference between the backbone atoms of the atom and the crystal structure of the compounds. So in this, the maximum, the preferred Q7 moieties, this may be a heat moieties, will be going for the dose screening, finalization and route of administration. Identified heat compounds may be subjected for different studies like pharmacokinetic studies and more pharmacodynamic investigations. And drug combination studies are also going to be one of the prospecta we can explore this moiety further. And this identified compounds where future modification with wide variety of substitutions can lead to get a very good moieties. Publication details already I have discussed. This is the first publication on diabetes, introduction and the literature review. Publication 2 based on the NT series and Q series. And these are the allied publications associated. And conferences attended. This is the second prize I got in Chandigarh group of institutions. Oral presentation. Thank you very much.
So, questions which me and one of the examiner for your PhD thesis, he also wants to know about. So first question is, justify a reason for selecting PAA and IAG enzymes as a target.
So in diabetes, evaluated glucose levels is the main parameter. In this, the two enzymes are very commonly preferred. What is alpha amylase and alpha glucosidase. These are the very good potential moieties. Even we have metformin, which is not very clear for this mechanism of action, but they used to predict that this metformin is going to be lower this, and even we have a drug like acarbose, maglitol, oglibose. So these drugs are mainly targeting on alpha amylase and alpha glucosidase. So we preferred due to the main reason for the anti-diabetic activity.
So you have described few compounds in series 1, that is
NT series, sir.
NT series. उसमें था NT17 है कौन सा कंपाउंड?
17 and 7 are.
और QT में?
QT7 and QT8.
QT7 है. तो QT7 NT17 और QT8, what are the structural modifications are required to make it more potent anti-diabetic agent?
Sir, generally, as per our study states that on C substitution, naphthalene and quinoline, these two are preferred as a clovengenol point of view, clovengenol condensation. On N substitution, we have done some modifications, right? So in N substitution, we observed the aryl substitutions are having a good potential as compared to the alkyl and haloalkyl. So on aryl group, we can prefer any heterocyclic group. Even we have done N18, which is pyridine. We have seen somewhat very good potential, but we have to do some modification substitutions on this preferring heterocyclic group, then it will be very good. So the best example is NT7, which is having heterocyclic group.
So you also conducted or performed antioxidant activity?
Yes, sir.
So what is the reason for this?
So in my presentation earlier I have clarified the point is ROS, oxidative, what we call the oxidation induced diabetes is one of the complication which is associated to the oxidation parameter. So we evaluated for that, the main reason. ROS.
So these were the questions from one. So you have synthesized thiazole derivatives. What are the common methods of synthesizing this?
Sir, for core thiazolidone, chloroacetoacetic acid and primary amines are generally used. And even in my introduction, I have reported 5 main important thiazolidone synthesis derivatives, like even microwave-induced method. More commonly, by using amines and chloroacetic acid is used for the synthesis of core thiazolidone.
So you used any solvent also?
Sir, I have no idea, sir, exactly. There is a solvent, sir. There is a solvents preferred in this synthesis.
Have you used any solvent?
In my synthesis, sir, I have used, for first step, I have used the methanol. In the second step, I have used ethanol.
इसको कैसे ड्राई करते हो?
Sir, recrystallization. One is रोटा evaporator से हमें solvent को एक्सेप्ट कर रहे थे.
So can molecular docking studies provide insights into the binding interactions between thiazole derivatives and target enzymes?
Sir, I'm unable to get the point.
Molecular docking studies आपने की हैं,
Sir,
वो कोई हमें इंसाइट्स देता है कि बाइंडिंग इंटरेक्शन किस तरह की होती है? जब मोलेकुलर डॉकिंग करते हैं
yes, sir.
तो बाइंडिंग इंटरेक्शंस किस तरह की हैं, कुछ उसके बारे में वो
Sir,
हमें.
we used to have pi-pi interactions, weak van der Waals forces, and hydrogen bonds. As when we see QT series, we have 4 hydrogen bonds observed in this QT8.
तो ये जो आप सारी इसमें आपने सिंथेटिक वर्क किया है या PhD आपने आज कंप्लीट कर लेनी है, इसमें कोई आपको मुश्किल आई है? क्या क्या मुश्किलें हैं?
Sir, initially,
क्या आपको लगता है कि जो आपके जूनियर्स हैं या जो फेलो कलीग्स हैं या स्टूडेंट्स हैं, उनको किस तरह से वो चीज़ें, पहले कौन कौन सी आपको hurdles आई?
Sir, hurdles is number one is, every faculty in LPU, of course I am also faculty, they are very known about my point is, एक बात बोलते कि शरीफ को लेते तो यहाँ से चले जाते. शरीफ सर को सुपरवाइजर, multiple supervisors.
Maximum, highest record.
So I have, sir is my final supervisor.
फाइनल मतलब कितना नंबर?
Sir, 6.
से अच्छा, जो आपके
जो भी सुपरवाइजर गए, they are well settled in government
They
job.
left LPU.
they left LP,
They are left LPU and they are settled in government job.
they are settled in
Government job.
Next is
Sniper.
गवर्नमेंट जॉब्स में चले गए सर सारे.
government job में. Who was your first supervisor?
My first supervisor is Gopal Tatak sir, and after that Narag sir, after that Nayak sir, उसके बाद अमित मित्तल सर गए और आगे भी, and last is
So he is not so lucky.
sir, I am very fortunate.
You are fortunate कि ये गए नहीं. Okay, so आपका कहना है कि जो आगे स्टूडेंट्स आए उनको एक परमानेंट सुपरवाइजर,
Yes sir.
जो आपका ड्यूरिंग योर सिंथेसिस, वो आपको एक
रिसर्च वर्क के बारे में बताओ.
सर आप वायरस रिसर्च वर्क के बारे में पूछ रहे थे, वो वायरस दूसरे जगह पे.
हाँ नहीं, मैं आपको जनरली पूछना चाहता था. चलो ये तो एक आपको एक फोकस तो था ना, आपने क्या
Sir,
करना
in
है?
research point of
During
view-
research आपको कभी कोई hurdles महसूस
Sir,
हुई?
in research point of view, जैसा supervisors are changing, my topics are going to change.
Your topics are
First, actually, I have started with biogonide derivatives. Then I have worked for 2-3 years at the time of Corona. Then one issue is Corona, and second issue is the chemicals are not going to procure.
chemicals. हाँ, yes.
That is
यही तो
actually
मैं-
they have spent maximum 4 years on this literature, and then
chemicals procure नहीं होते.
नहीं हो रहा था सर वो टाइम पे। यहाँ पे तो होता है, ऐसा नहीं है, बट एट द टाइम एट द कोरोना आई हैव स्टार्टेड माय जर्नी इन 2019 सर। एट द टाइम कोरोना आ गया, दैट्स ऑल।
During Corona,
But here I get
there are
maximum.
some unavoidable circumstances you can't get.
That is one.
Otherwise, you have all the facilities of
Excellent facilities, sir.
characterization.
For synthesis point of view, I have a special labs, okay, and even of course there is one thing, NMR is not available in campus. I used to prefer PTU.
So from where you did your NMRs?
NMR is from PTU, Punjab
Sorry?
University, Punjab University.
PU.
PU.
Okay, Chandigarh.
Chandigarh University, and some samples. Actually, I am from basically from Hyderabad. JN2 Hyderabad is my M.Pharm. Okay, there I have
There you
some
have.
contacts.
So, what is the future scope of your work?
Sir, of course, my work, when you're concerned about QT8 and QT17, so in future we are planning for the theoretical studies in animal preclinical studies. There we can get the clear information that how it is effective in the animals. Then further we'll go with good pharmacokinetics.
So yes, Mr. Sharfuddin, you did the presentation well also, and you answered all the queries, questions asked by me, asked by one of my fellow colleagues, and all your queries, all your answers are very much satisfactory. So I wish you a best of luck, and I recommend for your PhD. And also to congratulations and best wishes to your supervisor and co-supervisor under whose supervision you did